Welcome everyone. Today we are diving into the fascinating world of heredity. Heredity refers to the passing down of traits from parents to offspring. These traits are controlled by genes, which are the fundamental units of heredity found in DNA. Heredity means the transmission of traits from parents to offspring. Now let's see about genes and variations. Variations arise usually during the process of sexual reproduction. They may be few in asexual reproduction but many in case of sexual reproduction. The minor variations arising during sexual reproduction are caused by slight inaccuracies in DNA copying. In sexual reproduction, variations are also caused by crossing over process during meiosis. Beneficial variations helps the species to survive better in the environment. Nature selects the beneficial variations, therefore leading to evolution. Let's go to the next slide. Let's see about mechanism of variations. As we explore heredity, it's crucial to understand how variations accumulate. Asexual and sexual reproduction play a role here. Asexual reproduction generates limited variation, while sexual reproduction maximizes diversity through inherited traits. Let's go to the next slide. Now let's see about understanding inherited traits. Let's dive deeper into inherited traits. Humans display various traits from eye color to earlobes. Activity 8.1 demonstrated how traits like earlobe type might be inherited. This illustrates the diversity inherited from our parents. Let's go to the next slide. Now let's see about the rules of inheritance, Mendel's contribution. Now let's revisit George Mendel's groundbreaking experiment with pea plants. His observations led to Mendel's laws, defining dominant and recessive traits. These laws laid the foundation for our understanding of inheritance. George John Mendel started his experiments on plant breeding and hybridization. He proposed the laws of inheritance in living beings. Mendel was known as the father of genetics. Plants selected by Mendel was Piscium sativum, garden pea. Mendel used a number of variety garden pea to study the inheritance of 7 pairs of contrasting characters. Mendel conducted a series of experiments in which he cross-pollinated plants to study the character at once. Based on above monohybrid cross, he proposed law of dominance, which states that when parents having pure contrasting characters are crossed, then only one character expresses itself in F1 generation. This character is the dominant character, and the character which cannot express itself is called recessive character. The homozygous dominant trait is denoted by 2 capital letters, whereas the homozygous recessive trait is denoted by 2 small letters. Alleles are alternative forms of genes. For example, the gene for eye color has several allies. 2 major allies are brown and blue. Dominant ally expresses itself in the presence or absence of a recessive trait, whereas a recessive ally is able to express itself only in the absence of a dominant trait. Now let's go to the next slide. So now let's see about Mendel's experiments. As I have told in the previous slide about Mendel's experiment, now let's see about law of segregation. It states that every individual possesses a pair of alleles for a particular trait. During gamete formation, a gamete receives only one trait from the following alleles. A particular trait can be dominant or recessive in a particular generation. It states law of independent assortment. It states that alleles of different characters separates from each other during gamete formation. Genes carry information for producing proteins, which in turn control the various body characteristics. For a particular trait, the offspring receives one allele from the father and one allele from the mother. The combination of the male and female germ cells gives a diploid zygote. Thus, the normal diploid number of chromosomes in the offspring is restored. Different mechanisms are used for sex determination in different species. Now let's go to the next slide. Now let's see what is sex determination. The process by which sex of a newborn individual is determined is called sex determination. Sex chromosomes. In human beings, there are 23 pairs of chromosomes. Out of these, 22 chromosome pairs are called autosomes, and the last pair of chromosome which helps in deciding sex of the individual is called sex chromosome. Autosomes are a pair of chromosomes that are identical in appearance and are not involved in sex determination. Sex chromosomes are a pair of chromosomes involved in sex determination and are not identical in appearance. Sex determination in human beings. A male has one X and Y sex chromosomes, while a female has two X sex chromosomes. An egg fertilized by X carrying sperm results into a girl child. An egg fertilized by Y carrying sperm results into a boy child. The sex of the child is determined by the type of sperm that fuses with the egg. Acquired traits. For example, low weight of beetles. Inherited traits. Example, color of eyes and hair, shape of nose and earlobes. Now let's go to the next slide. Now let's see about the practical application and summary. Wrapping up, hereditary isn't just theoretical, it's practical and pivotal for survival advantages and evolutionary processes. By understanding hereditary, we gain insights into how traits are inherited, leading to variations and ultimately evolution. That's all for today's class. I hope you all like the video. Please feel free to subscribe and like the video. Thank you.